Liquid chromatography from Group 2. Our team members are Mara, Anna, Ely, and Nazrun. Operating principle.
Basically, separation happens due to differential partitioning between two phases. The stationary phase stays inside the column, while the mobile phase is the liquid that flows through and carries the sample. Components that interact more with the stationary phase move slower, while those that prefer the mobile phase move faster. That's how the mixture gets separated.
Next, main components.
So basically, this slide shows how an HPLC system works. The solvent starts in the reservoir, goes through the degasser and pump, and then the sample is injected into the system. Inside the column, the sample gets separated. After that, the detector detects each component and sends the data to the computer for analysis. For this slide, the table shows the function of each HPLC component. The solvent reservoir stores the mobile phase, while the degasser removes gases to prevent bubbles. The pump moves the solvent through the system at high pressure. The sample injector adds the sample, the column is where separation happens, and the detector detects the compounds. Finally, the data system records the signal and produces the chromatogram for analysis.
Next, let's take a look on application of liquid chromatography in chemical analysis.
First is the determination of chemical compound purity. To separate the main compound from impurities, reaction byproducts, or degradation products. Next is the analysis of active pharmaceutical ingredients, or API. To identify and quantify the active ingredient in drugs, and to detect impurities and degradation products. Lastly, there is pesticide detection. To analyze pesticide residues in food, water, and soil at very low concentrations. Since most pesticides are non-volatile and heat-sensitive, LC is more effective than other methods.
Demonstration of usage.
To understand HPLC, let's look at column chromatography. First, the column is packed with a solid stationary phase, and subsequently saturated with the solvent/eluent, mobile phase. Upon loading the sample, the stopcock is opened to initiate flow, while eluent is added continuously to elute the sample through the column. The separation of distinct colored bands is observed. The eluate were collected into test tubes as the first band exits the column. Collection vessels are changed for each subsequent band, until all components have been fully isolated. How to conduct HPLC. The HPLC modules were powered on, and the mobile phase is prepared. The analytical column then installed, and the system is equilibrated to establish a stable baseline. The analytical method, parameters, and sample sequence were configured. Analysis would commences upon initiation of the sequence. Data is acquired by the detector and processed as chromatograms. Following analysis, the column were flushed, the system is shut down, and the column is stored. Finally, the conclusion for this instrument. HPLC turns a complex mixture into individual measurable components. The success of the analysis in this instrument depends entirely on the tug-of-war between the mobile phase speed and the stationary phase retention. In order to attain a high-purity results and accurate data for better scientific decision-making.